Well, hello. Here's a PowerPoint signal. Let's see if it actually, there it is. Yay. Welcome to part 2 of 2.7. I am playing around with making this 3 parts just because this is such a huge concept. Again, some of you have different exposure on transcription, translation, and replication. Okay, in our last lecture, and then today in class, it is a gloomy Friday for future people. We were looking at DNA replication, right? So how DNA makes an exact copy via semi-conservative conservation. And now we're looking at how do we get from, oh my gosh, please, there it is. Okay. How do we get from here to there, right? So how do we, oh my gosh, all the things. How do we get from being these little pieces of DNA to having all these different proteins? And here we go. Yay, part 2. Transcription. So I know I've said this a couple times, but remember the word scribe, right? So even like a scribe in a courtroom, or in the medieval days, the scribe was the person that wrote things down, right? And we're moving things, right? So trans across. So we're writing down literally DNA from the nucleus and we're moving it to the cytoplasm to make that protein, right? So trying to remember that those two terms, because replication, transcription, translation all kind of sound the same. Okay, all right, let's start out with this process. More vocab, and a lot of it sounds the same, so we will try and do some practice in class with this. All right, okay, so the purpose, why transcription takes place. So you've got your nucleus, you've got your DNA, it's locked in there, so all the directions are locked in the nucleus, it can't move. So there has to be some kind of mechanism to take those instructions and move it to the place where it's actually built, right? So it has to be able to move around. And mRNA, messenger RNA, is the vehicle in which that happens. Okay, so the point of it is to synthesize an RNA sequence from a DNA template. There's a couple things we need to know about this. First of all, like only one strand is copied. I was trying to find things to help you guys see, right? Like, so this is what I found. So I'm going to use these pipe cleaners to help kind of show you, and we'll model this more in class. Okay. All right. So some tricky things. So first of all, the RNA is only copying just a small portion of the DNA. Okay. We need an enzyme to break the DNA molecule apart. In replication, that was helicase, right? And polymerase came through and added those bases. In transcription, RNA polymerase does both. Okay, so the RNA polymerase separates the strand and it makes the RNA copy, right? So helicase, there's no helicase leaving the nucleus, right? In mRNA. And there's only just a small portion that is copied. Like I'm just taking the directions for that one single protein that my body needs, all right? And after transcription, that RNA is released, the DNA strand reforms, and the RNA strand goes to the cytoplasm, all right, where we get to learn all these fun things like transfer RNA and ribosomal RNA, so many different types of RNA. Okay, so here's another visual of what that looks like. I will press play with the fun music. Oh, except for Miss Mars is blocking. So adding just that portion, DNA goes back together, the RNA is now formed, and I'm sorry, I'm trying to turn, oh my gosh, here it is. Okay, ready? And there it is. Okay, so I've got just that portion, right? So I've got my two strands like this, they're twisted together. Just the part that needs to be copied opens up, so it's not like the entire strand comes apart. Just that part that opens up gets copied, and then it moves to the cytoplasm where these other things happen. Okay, so that's my first part of it. Oh my gosh, I'm just always in the way. Okay, all right, so a little bit more about polymerase and having this visual here for you. The polymerase, it's called that because that's the type of molecule it is. And you just need to know that it does both, okay? So this little enzyme right here, this is my polymerase. And its job, again, is it breaks those hydrogen bonds, just like the DNA helicase does, right? So the RNA polymerase does the same thing. It breaks it. And then it makes those covalent bonds on just this single strand, right? So it separates the DNA, makes the RNA copy, and then it's released and the DNA comes back together. Okay, so that's the more visual part of that RNA polymerase. There's some more enzymes we'll learn about here coming up too. Okay, So some vocabulary for that strand. So if I have my strand, right, that's coming together, you're not gonna be able to see this. I don't know why I'm doing this. Okay, so you've got your red and your black strands here, right, and they're coming together. So I'm just need to copy like this small little portion. So we already talked about that it opens it up and transcribes just the nucleotides, but there's two sides of that DNA strand, right? So it's like, how does it know which side it's going to copy? Because it's those corresponding bases. And so the antisense strand is the one that is actually copied down, right? So the antisense, the sense is not transcribed. Literally, this doesn't make sense to me. It's like, why would that doesn't make sense? The antisense is the one that's transcribed, right? And so if I pulled these strands apart right here, you've got them exposed, right? So if this is my antisense strand, the black one, that's the one that's going to be copied. The red part, this is my sense strand, is not it's not copied, right? It has those corresponding. Right? We'll talk a little bit more about like, how does it know? Okay. So this sequence is identical to this transcript except for the TU. We'll talk about those bases. So if here's my antisense strand, right? Here's my sense strand. Then my transcript, right, where I'm writing it down, is going to be the one right here. So T pairs with an A pairs with a U. Oh, I should probably, here, hold on. Let's go like this. In our base pairing rules, remember outside of the nucleus, right? And we'll do tons of practice with this. So inside the nucleus of my DNA, my A pairs with my T, my G pairs with my C, right? My C pairs with my G. So that's the complementary strand, and my T pairs with my A, all right? But remember that messenger RNA during transcription does not have, right? So thymine is not able to leave the nucleus. So when I'm transcribing this strand right here, right? So my A would then now pair with a U. My G still pairs with a C. My C still pairs with a G. And T will still pair with an A. So there's still adenine outside, right? And so my transcribed strand has the U base in it instead of the T. That's something that we will do a lot of practice with because that's just something that you have to practice. Okay. All right. So after we have my sense and my antisense strand copied, oh, there we go. It moves on to translation. Okay, I'm just going to keep going, but I might break this up. Okay, so part 3, translation. So now I have this written down in this code, right? And now I'm going to move it on to my ribosome where the protein is actually made. Okay, so part 3, translation. Whew. All right, so now we're going to my ribosome. So my messenger RNAs, I've got this written down. I'm going to go to my ribosome because that is the place where proteins are made. We learned that all the way back in topic 1. Now we're going to learn around why, okay? This translation is where it actually takes those bases and translates it into an amino acid, right? So there's no more just copying. I actually have to make something else. You can think of like translating it into another language, right? I'm not just copying it down. I've got to make it into something else. That involves polypeptide synthesis, polypeptide. What did we learn about peptide bonds, right? By the ribosome. So we're making proteins. We've already done all of our protein bonding, right? And so it takes those different nitrogen bases and it's putting them together, right? So my amino acids are being linked together to make a protein, okay? Those peptides are encoded by the genes. The genes have the directions. The mRNA strand leaves the nucleus, and then it goes to the ribosome to be translated and linked into proteins. Here is another visual of that. Sorry. Oh, links, see? Okay. Polypeptide. And we're not going to watch it again. We can watch it again in class, all right? So now it's going together to create the protein. Okay, so step-by-step translations of the, process of translation. Okay, so there's actually a mnemonic device coming out, this overview. So I will have you copy this down, maybe just the MR cat, and then we will fill it in class, but I'll talk about that in a second. Okay, so let's go back and we've got here, right? So we've got my mRNA strand here. This is the one that came from the nucleus, right? So here's my nucleus, yay. and it's locked up. The DNA is locked up. We talked about it has those nuclear pores. My mRNA copies just a little strand, and it travels out here to my ribosome, right? So my ribosome is out in the cytoplasm, or it's on the ER, right? So the rough ER has those ribosomes attached, okay? All right, so that's my messenger RNA. My transfer RNA is this little molecule here, right? And this transfer RNA has an amino acid attached to the top of it, okay? So the transfer RNA still has these bases, right? So it has my nitrogenous bases, and depending on the code from the RNA, it matches up with the corresponding bases, right? A with U, G with A, right? So I'm sorry, no T, sorry. G with C, right? And so it matches up there, and then it drops off the amino acid, makes the polypeptide bond, and then moves on, right? So messenger RNA goes there, and then it starts to read in sequences we call triplets or codons. We are going to do a ton of work in class with codons and using that chart. So for now, know that a codon is 3 bases, and the anticodon is the one that goes with it. So much vocabulary, okay? So my transfer RNA transfers the amino acid, and it goes with the corresponding or complementary anticodon. So anticodon with a codon. And then the ribosome actually moves along and joins them together, and then it's released. So when that code is done, when everything's been copied and translated, remember we talked about that it's a temporary little directions. It breaks itself apart, and it goes, breaks itself down, and then is reused, right? Sent to get more or used somewhere else in the cell, okay? So for notes today, just go ahead and write down in your margins, right? So write down in the margin, Mr. Cat app, right? So write down Mr. Cat app, but don't fill it in right now, right? So don't fill in this right here. Don't fill this in. You should block it, right? Just write this down in your margins. So Mr. Cat app, because this is kind of a cool way to remember the order, but also the story. So it's not just a mnemonic device. It's like a story about how the messenger RNA gets to the protein. Okay. All right. The other thing that we'll be doing in class is looking at, this, I'm going to go back in just a second. Is looking at this chart right here. And doing a lot of practice for making those different amino acids. Okay, so let's talk really quick about the genetic code. This is the last part. The genetic code are the rules, right? Like the central dogma by which my mRNA strand goes into my protein, okay? These rules, right, have already been outlined. My codons are triplets. They correspond within a particular amino acid, right? So the amino acids are coded for both by those specific bases. The order of them determines the order of the amino acid and thus the protein, right? So it's the order of the mRNA that matches the tRNA that carries the amino acid that makes the protein, right? It has to go in those order, and those are the kind of rules of the genetic code. Some key features, so some things that are universal, and then degeneracy, I'll talk about in a second. But universality means like all of these codons, amino acids, they are the same in all organisms except for just a couple viruses. But for the most part, the AUG in one organism codes for the same exact amino acid in another organism. So this is a universal genetic code. So the amino acids that are made in one organism are made in the other, right? It's not like each one has a specific key, okay? The theory or the concept of degeneracy is the idea that more than one codon may code for the same amino acid. I'm going to show you that in the next slide, so that makes more sense, right? There are a total 64 codons, but only 20 amino acids, right? So some of those codons code for the same amino acid, and that allows for something called silent mutations, and we will look at that during our DNA Gone Wrong unit. Okay, so that's the genetic code. All right, on this next slide here, do not copy down. Let me go here. Do not copy this down here. You can just look at this. This is a codon chart we will be looking at, but you should see right here, so look at all 4 of these codons code for the one amino acid, leucine, right? These 2 code for histamine, these ones, so some of them are very specific. I want you to pay attention to these ones that are the stop codes, because that's a really important, that's where it stops. And then there's also start codons, right? So we'll look at those too. So the genetic code of messenger RNA, it starts with a start, yay, vocab that makes sense, yay, and it terminates with a stop, All right. This start codon AUG. So, but you could see how if something is mutated or put out of order, all of a sudden I have a very short protein or starting someplace that it shouldn't, right? The degeneracy always occurs in the third base. So that is the idea that some of them come. So if you see right here, CCC, UUU. So this one right here, that third base right here, that is where we see the differences. So the first two are always the same, and the degeneracy occurs in the third. The reason why, why that like scientists believe that maybe that's possible is called the wobble hypothesis. And it's just trying to show that like if these two link right here, right, that the shape of this transfer RNA for this last one, it's not optimal for base pairing at the third one. So it just kind of like whatever the first two are, that third one doesn't matter. It's gonna attach there. Okay. All right. Let's move on here. All right. The reading frame. So we already talked about these a little bit, but the triplets that are called codons. The reading frame. Here. I'm gonna go like this. You know what? Stop. I'm skipping the reading frame one. We just went through all of that. It's just kind of a summary. So, and this is also a summary. So, but this is actually a little bit more of an application, and this is where we'll end today. So because we do have this universality, right? So that the genetic code is the same in everything, we can actually use this to our advantage to create things and have like insulin. Right? So the protein insulin for people that are diabetic, not able to make it, we're actually, so if they have to do insulin injections, very long ago, I'm saying not very long ago, 1950s, 60s, they used to actually like have to take them from cows and pigs and things like that. But now they're actually able through this process of recombinant plasma, like actually putting it into that specific code into a bacteria, they're able to mass produce insulin without using living organisms, right? And so this is how this process works. So I've got my human cell and I have this bacteria. We'll talk how bacteria is used a little bit with different types of DNA processes. So I just extract that gene, right? So I've got my portion of DNA that we know codes for insulin in all organisms, insulin in a cow, chicken, pig, like it's all the same, right? And then we take what's called the plasmid and put those together. So it actually cuts that little part. We'll look at some designer babies too. And it creates what's called a recombinant plasmid. So it takes this plasmid and it inserts that little genes. So we get a transgenic bacteria that is creating that protein in mass production, right? So it's just creating that insulin protein, and it's grown in cultures and labs, and then they're able to extract insulin, right? So using this process to create good. Recombinant DNA is also used in kind of controversial things. Again, we'll look at that in class, but the idea that we can cut out genes and insert genes into different organisms, as you can see, like that creates a lot of opportunities, right? Good and bad. So that is looking at the universal code in action. Okay, so in class, we'll do a little bit of summary, but that's it, right? That's it. So this is our part one, right? We've already gone through these in part one, and this is our part two, all right? Tons of practice to be had with looking at those codons and whatnot, so we will take some time in class to do that, all right? You guys have a fabulous weekend. I will see you later.